In this section of the lecture, we'll talk about the transport mechanisms that exist at the cell membrane. You could describe the cell membrane in a couple of ways. We already talked about fluid mosaic model, bilipid layer. Now we will talk about semi-permeable membrane or selectively permeable membrane, because the cell membrane chooses what should be transported and what should not be transported. How it does that is going to be something we will focus on in this lecture. Okay, first of all, let's organize the transport mechanisms into passive and active types. Passive transport mechanism is where you do not use ATP, you do not use energy. Okay, ATP is the most common energy currency in the body. All right, so if the movement of a substance is from a higher to a lower concentration, where there is a concentration gradient, it's a natural process and you don't need to use ATP. Such transport mechanisms are diffusion, facilitated diffusion, and osmosis. Active transport mechanism is the exact opposite. Here, the movement is against concentration gradient. You are moving a substance from the lower concentration to that of its higher concentration. You see? So the concentration is already high and you're still moving more substance into that area. Okay, that's where it's like pumping action, and the pump requires energy to work, and that's where you have to use ATP. Okay, active transport is the main type of transport mechanism that depends on ATP. You will soon learn that there are two types of active transport. One is primary active transport, where the ATP is needed directly in the process, whereas secondary active transport is where energy is used, but it's not actually in the movement of the substance that has to be transported, but elsewhere. Okay, so it's an indirect use of ATP. Yet another type of transport mechanism that exists is vesicular transport. Vesicular transport is where the substance is not moving through the membrane, but the membrane would go through modifications. It would fold, it would assume this cup shape, and then the cup ends would meet in the place where the two edges meet will get chipped off, and then that's how a substance is transported. Do we need ATP for this? Well, again, it's a function that eventually will require the synthesis of the membrane to be used to form this vesicle, but not directly. So it is not like it's dependent on ATP at the moment, but you would use the source of energy to construct various things that are involved in the vesicular transport. So that's where the question mark is. All right, so with this, we will focus on each and every type of transport mechanism. The first one happens to be simple diffusion. Okay, simple diffusion is where you're looking at three situations. Here is the membrane. Okay, it is permeable to a substance that's in larger concentrations outside the cell and in lesser concentrations inside. In this situation, there is just like nothing inside, a lot outside. Okay, over time, this is what happens. As you allow the substance to move, what happens is it gets into the cell slowly and steadily. And what has happened here is that the concentration is more or less the same, both outside and inside. This is where you call net movement is zero, because there may be molecules moving both ways. So the net movement would be zero. Here it's the movement from outside to the inside. Okay. Because it's from a higher concentration to a lower concentration, this is what we call as simple diffusion. Simple diffusion is when it occurs across a cell membrane that's permeable to this particular solute. So you could imagine this as a glucose molecule or just a drop of ink that was dropped outside the cell, and how it would make its way into the cell as long as the membrane is permeable to this molecule. Okay, so no energy needed, and the movement would occur until an equilibrium is reached. Well, here we want to understand various factors that would affect the rate of diffusion. That's what fixed law of diffusion deals with. A number of factors that would impact the rate of diffusion. Okay, some are directly involved, some are inversely related. Okay, so let's look at concentration gradient. Okay, as long as there is concentration gradient, from one place is higher concentration and another place is lower concentration. Greater the concentration gradient, greater the rate of diffusion. Do you agree? If there is a small difference, then the rate at which diffusion happens probably is slower than when there is a greater concentration gradient. That same thing applies to pressure gradient. Higher pressure to lower pressure. We will use a lot of examples, especially in the blood vessel physiology or respiratory physiology. We'll talk about pressure differences and how that would help in the movement of substances from higher to lower pressure. Size of the molecule. Well, if the molecule is smaller, you see, it can move rather quickly than a larger molecule. Don't you agree? So the size matters. Okay, smaller size, greater rate of diffusion. Larger size, lesser rate of diffusion. Electrical charge. Well, depends on the membrane charge and the substance to be transported. Okay, if the solute has the same charge as the membrane does, okay, we already know that phospholipid has this negative charge, and then if you have a solute with a negative charge, it may not be able to move through because of the same charges. Opposite charge, on the other hand, can get through more readily. All right. Now let's talk about thickness of the membrane. Okay, more thicker the membrane is, less diffusion there is. Imagine the lung in conditions such as pneumonia. There is a lot of secretion, and this coats the alveolus, and therefore the wall is thicker, and the gas transport mechanism, gas exchange, would be less compared to when you have healthy lung tissue, correct? And then distance a molecule has to travel. Well, if a substance is to travel a short distance, diffusion would be faster. If the substance has to travel a long distance, then there is less chance of a substance crossing the membrane in a greater amounts. So greater the distance, lesser the diffusion rate, and vice versa. Temperature. Then here, heat helps with the diffusion. If you increase the temperature, then there is greater diffusion. Exercise is one good example. When you're exercising, gas exchange would be much greater. All right, a fever is another condition where you'll be exchanging more gases than at other times. Good. So now we will come back to the discussion of pores and channels. Here, what we're talking about is diffusion using protein structures. Okay, these are water-soluble molecules and may not be able to get through the membrane structure. Remember, membrane structure is all lipid, mostly lipid. So now you have to depend on protein structures. We looked at the proteins before, integral proteins. The shape of the protein is such that they can create pores. So that's where you call them as pores. Or channels. Channels have gates, pores do not. Okay, pores always open. Pores can be selective. They can be selective to just water. They can be selective to just ions. Even among the ions, it could be just the potassium ion that they allow in a certain amounts. Okay, channels, as I mentioned, are gated, meaning they are selectively permeable. Okay, they are programmed to move only one specific solute molecule. Okay, here you're talking about sodium channels, sodium potassium channel, etc. Okay, now you are looking at leakage channel. This is one type of channel. This is gated, but the gate opens randomly. You really don't have a way of explaining what makes the channel open or what makes the channel close. Here is an example of a channel being closed or a channel being randomly open. Okay, so now you're talking about the concentration here being greater than here, and if this substance has to be moved to an area of a lesser concentration, the channel may randomly open and allow this substance to move to an area of a lesser concentration. That's when you call it as leakage channel. Well, in the cell, you typically have quite a few potassium channels. These are leak channels. So a potassium ion is constantly leaving the cell because of the presence of leak channels. There are fewer leak channels for sodium ions in a typical cell. Okay, just to give you an example of a leak channel. All right, here is an example of voltage gated channels. Okay, what it is the channel opening or closing depends on a specific voltage. Voltage is the amount of current that's in there. Okay, in here, the example that you see has a certain voltage, -70 millivolts. You will hear this number a lot, and that is a typical neuron cell membrane potential. Okay, normal resting membrane potential, -70. Anyway, so in a normal situation, what do we see? The channel is closed. But when the voltage becomes less negative, it's -50 now, what has happened? The channel becomes open. Okay, and allows the movement of a certain ion. So this is voltage dependent type of channel. That's what we learn. Okay, we will look at specific examples in later chapters. Okay, with this we'll talk about ligand gated channels. Very important type of channel again. What it is this channel here that you're looking at, okay, has a binding site. Do you see that? And this binding site is supposed to bind to a specific substance. You call it as the ligand. This ligand could be a drug, this ligand could be a neurotransmitter, a hormone, etc. Okay, so here a specific substance binding to the channel. All right. Right now, in this example, you do not have the actual binding. There is this molecule called acetylcholine, you see, but it's not bound to the channel yet. So the gate is closed. What happens here? the substance is actually bound to the protein, that would change the configuration of the protein. Something happens here, the protein structure gets altered. Now the new shape of the protein allows this opening, and now ions would move in a different directions. You're looking at certain ions moving out, certain ions moving in. The details of where you would find these channels and how they work and what that means will all be discussed at a later time. But this is ligand-gated channel. What do we learn from ligand-gated channel? This is programmed by the presence or absence of a specific ligand. So if I am looking at this ligand-gated channel, if this is not acetylcholine, but let's say another neurotransmitter like serotonin, okay, would this channel open? Maybe not, because this may not recognize that particular neurotransmitter. Okay, so a specific ligand is what is needed to open the channel. Ligand gated, that is, channel opening depends on a ligand. So there are different types of channels, and we looked at some examples: leak channels, voltage gated channels, ligand gated channels. The important ones are mentioned here, and we are going to be learning about them in a greater detail elsewhere. Okay, so that's all about diffusion, and now we are talking about channels. That was a nice segue for us to understand facilitated diffusion. What is facilitated diffusion? It is diffusion that is done with the help of carrier proteins. Why? The solid molecule is too big to move by itself. It needs the help of a certain protein. So you're looking at the functions of proteins, membrane proteins here. We already looked at the channel function. We already looked at the pore function. This time you're looking at the carrier protein function of membrane proteins. Well, how does it work? Okay, this is again the movement of solute from a higher concentration to a lower concentration. The green substance here, whatever it is, we can make up a name for it, okay? Typical substances that are transported by this type of transport mechanism is glucose and amino acids. So maybe we could call these as glucose molecules. They are in large amounts outside of the cell, extracellular fluid, and in smaller amounts inside. So there is this concentration gradient. Okay, there is a carrier protein in the membrane. Okay, now this carrier protein is sensitive to the type of solute it transports. So basically what that means is it cannot transport any substance and every substance, but it is very selective. So what happens is this substance, glucose in our example, binds to the protein. What happens then is the structural modification of the protein would allow this space to become wider. Did you see? Now there is room for it to be brought into the cell. That's it. It's the binding of the substance that modifies the shape of the protein, which we call as carrier protein, that moves the substance into the cell. So whichever way it moves, the substance, it doesn't matter. You understand the mechanism. You need the help of carrier protein for diffusion to happen. And that's what we call as facilitated diffusion. Okay, so look at some other characteristic features of facilitated diffusion. What you're looking at here is the concentration gradient. There is that concentration gradient. Now, what happens is simple diffusion, if the membrane is able to handle simple diffusion, that can go on in an endless manner. As long as the concentration gradient exists, simple diffusion will occur, okay, forever. But what you see here is a limit of transport for facilitated diffusion. Okay, facilitated diffusion, even though the concentration gradient exists, it can only transport so much, not as much as simple diffusion is. Okay, you know why? Because facilitated diffusion depends on carrier proteins. The number of carrier proteins that exist in the membrane dictates the maximum capacity the facilitated diffusion can occur. Get it? So that's how you call it as Vmax. Vmax is the maximum level, maximum amount facilitated diffusion can occur within a cell. Okay? Well, when you think about diffusion versus facilitated diffusion, because you have carrier proteins, once you have carrier protein available, the movement of a substance could be like really quick. Boom, 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 right? Now, what about when you don't have the carrier proteins? You are capping it. Okay, you cannot transport anymore. So that's what you call as transport maximum. Transport maximum is Tmax. Tmax applies to facilitated diffusion, but it does not apply to simple diffusion. Does it make sense? Yeah, good. With that, we have understood diffusion and facilitated diffusion. Now we can talk about osmosis. What is osmosis? Well, here you have to imagine the membrane to be not permeable to solute, but is permeable to water. Only in this situation, you would see water movement. Okay, so it's the movement of water. Osmosis is where you expect water to move. Now the question is, in which direction? Well, if you have two concentrations across the membrane, so here you're looking at lower concentration versus higher concentration, correct? So if water moves, which way would it move? Now think about it this way. When you have lower solid concentration, if water moves into this compartment, it would dilute this more. So water does not move into the area of lower concentration. When you have lower concentration, there is more water in here, agree? So water from its higher concentration to its lower concentration. When you are thinking about only water, okay, water from its higher concentration to lower concentration. How do you know that there is less water? Because there is more solute. Okay, it's taking more space, so there is less water. Agree? That's one way to look at it. Or if you're looking at it from the solute point of view, you have to reverse it. Okay? Water moves from lower solute concentration to higher solute concentration. Okay? It's important for you to separate water and solute here. If you're thinking about water, it is just like diffusion, higher to lower concentration. If you're thinking about solute, you have to reverse this. Okay, whatever it is, now what you see is water moves from lower solute concentration to higher solute concentration. Over time, you see that there is more water here, less water here. Okay, so there is that volume shift, but the concentration should be the same. So this is what we're looking at. So there is that equilibrium that has been achieved. Okay, so that is osmosis, movement of water across the membrane. We will talk about osmosis a lot throughout our course, so be prepared. Now we will just understand the definition of osmotic pressure. The force with which water moves from the area of lower solute concentration to the area of higher solute concentration. The force with which water moves is what you can measure, and that's what is called osmotic pressure. So greater the difference between the two compartments, like greater the concentration gradient, greater the osmotic pressure. Don't you agree? Yeah. So that is important for us to know. Osmotic pressure is determined by the number of particles per unit volume of fluid, not by the mass of the particles. It does not depend on the weight of the particles, but the amount of water that gets to move. Now another definition, osmolality or osmolarity. Osmolality and osmolarity in physiology are one and the same. So as long as you know that, that's all that matters. Okay, osmolality is where you're thinking about kilogram of water. Osmolarity is where you're thinking about liter of water, but realistically, because you're dealing with water, 1 liter of water is the same as 1 kilogram of water, so it does not matter to us. Okay, if it is chemistry where you're dealing with different fluids and the kilogram, liter are not one and the same, then you would run into to separating osmolality from osmolarity. For us, it's one and the same, as long as you know this. All right, now let's talk about units. 1 osmol is 1 gram molecular weight of osmotically active solute per kilogram of water. Okay, so that's the definition that's already laid out. Now, what I want you to know is the solute molecules, solutes that you deal with could be osmotically active or not. Glucose does not break apart any further than glucose. It remains as glucose. So 1 glucose, when you put it into solution, it remains as 1 glucose. So 1 gram molecular weight of glucose is 1 osmol. Get it? Of glucose. 1 osmol is 1 gram molecular weight. Okay? Now, if you think about another solute, this is now inorganic. This is organic. This is inorganic. Sodium chloride, 1 gram molecular weight. Okay, when you put it in a solution, what happens? It dissociates into sodium ion and chloride ion. So now you count them as 2 osmoles. You see? So the weight doesn't matter. In fact, when you think about the weight, glucose weighs more than sodium chloride does. You see? So weight does not matter. The mass does not matter. It is the ability of the solute to dissociate into more than just 1. Okay? So that's where the numbers can be confusing for you. Okay? Now we talked about osmolarity is the same as osmolality, so we will move on. Now, one other number that's very important for us is body fluids have an osmotic concentration. A certain osmotic concentration. How? Body fluids are not just pure water, okay? Water plus some other things. Okay, what do you find in extracellular fluid? Well, it depends on whether it's blood or interstitial fluid, right? And now we could also think about intracellular fluid, that is the fluid inside the cell. Okay, what is in it? Is it pure water? No, nowhere it's pure water. You see, there is always something else like ions and proteins or nutrients that are water soluble and they are dissolved in these fluids. So that's how you have a certain solute concentration along with water. And now, if you were to measure that, the normal osmotic concentration of body fluids, is approximately 300 milliosmol. 300 milliosmoles. Okay, whether it's interstitial fluid, whether it's blood or intracellular fluid, it's always going to be the same. Okay, whenever there is a shift in this osmotic concentration, water would move in one direction or another, ions would move. correcting the osmotic concentration to be this. Okay. Here is a detail of tonicity. Tonicity is basically looking at the concentrations of two different fluids. So here you're talking about cells being placed in one fluid condition or another. Okay, so here you're talking about isotonic condition. What that means is red blood cells are in the plasma that has 300 milliosmol concentration. Okay, what that means is these cells are going to be fine. That's the concentration inside the cell. That's the concentration outside the cell. That's when you define that as isotonic. Same concentration inside the cell as well as outside the cell. Net movement is going to be zero. Water going in, water coming out. So it's kind of, no gain, no loss type of situation. Cells are going to do well in this condition. Okay, what are we talking about here? This is hypotonic condition. Red blood cells happen to be in plasma that's not 300 milliosmol, but it is diluted. Diluted plasma. So make up a number. Maybe it's 200 milliosmol to 50 milliosmol, not 300 milliosmol. So what happens? Water, remember, would move from lower solute concentration to higher solute concentration. When you have 200 milliosmol here, 300 milliosmol here, this is where more solute is, that's inside the red blood cell. So water would enter the red blood cell. Cell begins to swell, can you tell? And the swelling can go on for a while, and at some point the cells may burst and die, and that's what you see in here. Hemolysis, it's the death of the red blood cell because of too much water inside. Okay, so that is hypotonic condition, and how water enters the cell, makes the cell swell and make these cells die. The opposite situation is hypertonic condition. Hypertonic is where you have red blood cells in a very concentrated solution. Okay, someone did some wrong calculation, maybe instead of 0.9% saline, maybe 9% saline, not good, but that could happen. And now we have very concentrated plasma, that means more solute here compared to the 300 milliosmol concentration inside the red blood cell. So the movement of water is from the cell towards the fluid here that we can assume as plasma. So it's now loss of fluid for these cells, and the cells begin to shrivel. Okay, shriveling is what you call as crenation. Okay, cell death because of the loss of water from the cell. So that would be the consequence for you to remember. When the solute concentration is greater, you would lose water and the cells may begin to die at some point. Okay. So here is the water moving out of the cell into the solution where the cells are present. Okay, good. So I think I'll stop at this point and talk about other details in another video.